We'll start with the session. We invite Prasani Yapon Posire to present BACIVAC, a novel RNAi vaccine from a non-pathogenic bacillus substellar cell expressing dsRNA EHP to protect the microsporidian infection in shrimp. You are given 15 minutes, 10 minutes for presentation, and 5 minutes for the question-answer round. And after 10 minutes, the bell will ring so that you know that it's now the 10 minutes.
Okay.
Thank
you for your introduction.
Thank you.
Okay. Good afternoon, everyone. My name is Prasani Posire. I come from Biotech Nasdaq, Thailand. Today, I'm pleased to share our results on the BACIVAC, a novel RNAi vaccine from the Bacillus subtilis expressing double-stranded RNA EHP to protect microsporidian infection in shrimp, specifically in Excitonicrospora hepatopeniae or EHP. EHP is a serious microsporidian parasite affecting shrimp aquaculture. The characteristic and significance of EHP have already been described in detail by Dr. Kalaya yesterday and Dr. Rajendra in this morning. Everyone knows the importance of EHP also had a significant economic impact. For the example, the economic loss. For the example, the economic loss about the 200 million US dollar in Thailand and about 567 million US dollar in India. Clearly, EHP is a major threat to shrimp production and the aquaculture industry. One of the key process in EHP infection is spore extrusion. Proteomic analysis of the extruded spore extract identified the polar tail protein 2 or PTP2. PTP2 was detected at a high abundance. Furthermore, the polar tail germination associated with PTP2 was observed in both of the hepatopancreas and the intestine, but not in the stomach. Based on this finding, PTP2 was selected as the targeted for subjects to investigate the role of EHP infection. The double-stranded RNA specifically target PTP2 was produced by in vitro transcription and injected into the shrimp. This treatment successfully suppressed EHP infection 21 days after the first double-stranded RNA injection. Double-stranded RNA is a key component that triggers RNA interference to suppress the target gene. There are several methods for producing double-stranded RNA, including in vitro transcription and in vivo bacterial expression. Here, we focus on using Bacillus subtilis as a cell factory for double-stranded RNA production. Why Bacillus subtilis? This bacterium is already widely used as a probiotic and is in the food production and is officially generally recognized as safe or GLAS. And it can function as an efficient double-stranded RNA biofactory and the potential for the low-cost production. In addition, this method is the potential for practical application in the shrimp farm. For our proof of concept, we select strain BGT22, which lack of the RNA3 activity. We construct a recombinant plasmid carrying the HPTP2 gene and introduce it into the Bacillus subtilis strain and culture the cell at 27 degrees Celsius for 12 to 16 hours, for 12 to 16 hours. After harvesting the cells, we extract the double-stranded RNA and found that it was digested by RNase-3 but not RNase-A. This double-stranded RNA is the sufficient quality for use in the shrimp injection. Many studies show that using double-stranded RNA shrimp reduce pathogen infection, such as virus, including white spot syndrome virus and the IHV, and also in the Mycosporidium EHP that I mentioned earlier. Based on this result, we began to consider another question. Does double-stranded RNA not only induce RNAi to suppress EHP transcription, but also prime the shrimp immune system in a vaccine-like manner? If immune priming occurs, can measurable difference in the shrimp immune response to be observed? This is the experimental design to answer the question. Shrimp were divided into the flow group, sodium chloride and double-stranded RNA EHP-TP2 only, and sodium chloride plus EHP and double-stranded RNA EHP-TP2 plus the EHP. Shrimp were injected with double-stranded RNA and challenged with the EHP by cohabitation with EHP-infected shrimp. The double-stranded RNA injection were given once every 7 days, and shrimp were sampled 21 days after the first double-stranded RNA injection. At that time, we collect hepatopancreas to measure EHP level and collect the hemolymph for the transcriptomic profiling. The result of this experiment found that RT-PCR show the group received double-stranded RNA and then challenged with the EHP. Subsequently had the low level of EHP-TP2 gene when you can see the group of the shrimp injected with the double-stranded EHP-TP2 plus the EHP show the low level of mRNA PTP2 when compared with the EHP. Similarly, quantitative PCR of the spore wall protein gene reveal a significant reduction in copy number. The most direct explanation for this decrease in EHP gene expression is that the double-stranded RNA trigger the RNA interference pathway, leading to degradation of the target mRNA. However, the question remains whether the other pathway might also contribute to this suppression. The transcriptomic analysis of the hemocytes reveal extensive change in gene expression. Genes that were downregulated are shown in the blue cluster, whereas those that were upregulated appear in the red cluster. This pattern can be seen in the comparison group between the sodium chloride versus EHP and sodium chloride versus double-stranded EHP-TP2 plus EHP and EHP versus double-stranded RNA-PTP2 challenged with the EHP. These are the sample of the transcriptomic response that observed in each comparison group. Across on group, there was clear activation of immune response signal, immune signaling response. Moreover, in the sodium chloride versus EHP comparison, you can see the change of the expression of a lot of the pathway, including putative cell repair pathway, neural signaling and genesis pathway. And also, you can see the mucin profile and melanization alteration. In addition, in the group of sodium chloride versus with the double-stranded RNA EHP-TP2 plus the EHP, you can see the differentiation of the gene expression, including angiogenesis, auto chitin degradation, melanization, mucin profile and chemotaxis. Furthermore, in the group EHP versus with the double-stranded RNA HPTP2 plus EHP, you can see the pathogen recognition, protease regulation, cell adhesion, and structural component. One of the most interesting genes identified in our transcriptomic analysis across all 4 experiment groups is endochitinase. This enzyme breaks down the chitin polymer from within the chain, cleaving internal bonds to produce chito-oligomers. And when we extract the DNA from the EHP, we use the chitinase and proteinase gene to destroy the spore wall protein, the spore of the EHP. In our study, the expression pattern of this gene was striking. In the sodium chloride control group, endochitinase expression was not detected in any of the 3 chimps. You can see in chimp number 1, 2, and 3. In the double-stranded RNA EHPTP2 only and EHP only group, expression appear in only 1 out of the 3 chimps, and you can see the low level of the this gene. Surprisingly, in the group injected with the double-stranded RNA EHPTP2 and then challenged with the EHP, expression was much higher, detected in much higher. You can see the 2 chimps, number 2 and number 3, show the high level of the endochitinase, and all of the chimps show the all expression. Even more remarkably, RT-PCR results from the group show that the level of EHPTP2 and the spore wall protein show the very low and almost equivalent to the control that didn't challenge with the EHP. These findings suggest that double-stranded RNA EHPTP2 not only trigger RNA interference against EHP, but also appear to stimulate the chimp immune system to produce defense level enzymes such as endochitinase from this result. This enzyme could help damage the chitin-rich spore wall EHP provide an additional mechanism to reduce infection. In other words, the double-stranded RNA EHPTP2 may act as a vaccine, priming the immune response to work in synergy with the RNAi to suppress the EHP. To summarize our findings, first, we achieved efficient double-stranded RNA production. Our engineer Bacillus subtilis produced about 700 nanograms of double-stranded RNA per OD. And next, we plan to further develop the Basivac platform to achieve higher double-stranded RNA yield and cost-effective downstream recovery, aiming for the practical application in commercial chimp farm. Second, we observe a vaccine-like effect of double-stranded RNA EHPTP2 transcriptomic analysis review immune and defense mechanism, defense response including upregulate and including upregulation of endochitinase expression. Next, we will investigate in more detail in immune pathway influenced by double-stranded RNA injection, looking at the timeline of response, the specificity of double-stranded RNA effect, and the mechanism of the double-stranded RNA uptake. This result demonstrates both the technical feasibility of double-stranded RNA production in Bacillus subtilis and the biological potential of double-stranded RNA EHPTP2 to prime chimp immune response. And now we currently analyze our large complex transcriptomic data set in detail, and we hope to discover additional pathway that could help explain the mechanism underlying the vaccine-like effect of double-stranded RNA. Finally, this work was funded by Medical Research Council and the International Science Partnership Fund and is supported by International Veterinary Vaccinology Network or IVVN. And I would like to express my sincere thanks to our collaborators, Dr. Jamie, Dr. Amy, Dr. Kalaya, Dr. Ponsupi, and Dr. Tewa, for make the project possible and working together with us. And last, but certainly not least, I want to thank the true hero. Without their hard work and dedication, our research could never have reached this point. Thank you very much. And finally, I would like to thank everyone here for listening to my presentation today. If you are interested in collaborating or would like to share the idea, you can contact me from this email. Thank you for your attention.
Thank you. I think we have time for one very quick question. No? Okay, well, thank you.
Thank you so much.
Thank you. Oh, no, sorry, I think we have a question. I think we do have one question, sorry.
So this year, double-stranded RNA, will it be effective against spores or only on active parasites?
Yeah?
Your double-stranded RNA, will it be effective against spores or only on active parasites?
For this experiment, I use the double-stranded RNA target EHP only.
Thank you.
Our next presenter will be Goklesh Kumar, presenting vaccine target discovery in a fish parasite using an in vivo model. 10 minutes for presentation. There will be bell at 10 minutes and then 5 minutes for discussion, please. Please keep up the time. Thank you.
Thank you, ma'am. Good afternoon, everybody. I am Goklesh Kumar from ICR, Center Marine Fisheries Institute, CIMO Farai, Kochi. We have done this work at the Badminton Uni, Vienna, Austria, and two young scholars from ICR Institute joined us, like Arun Sudagar and Saloni Shivam. And we have done this work under the supervision of Professor Ilmat Bawli. So I would like to talk to you about vaccine target discovery in a fish parasite using in vivo model. This parasite may be new for you. Here is the outline of my presentation. Overview of parasitic diseases, endoparasite, tetracapsuloid salmoni under the phylum of Myxozoa, and mysterious disease, PKD, bryozoans, and we applied in vivo induced antigen technology to identify vaccine target candidate for this endoparasite. As we are aware that parasite and organism live in, that is the endoparasite, or if on the ectoparasite, they depend on the host for food and causes some degree of injury. Parasites are a major serious concern for fish population as well as cultured fish, and Dr. P.K. Sahu also indicated that parasites are serious threat. And parasites such as Myxozoa, Protozoa, crustaceans, monogenians, and halvenes results a very highly economic losses in salmonid in aquaculture industry. And they have negative effect on the host, either direct damage or indirect damage. If we see the endoparasite and ectoparasite, they causes annual economic losses around 1.05 billion to 9.58 billion US dollar. So here I would like to focus only on endoparasite, which is called Tetracapsuloides bryosalmoni. Tetra means the parasite contain 4 capsules and maintain their life cycle in bryozoa and the salmonid host. So here we can see the diagram of this parasite having 4 capsules, and this parasite uses bryozoan as a primary host. And the life cycle of this parasite involves 2 hosts like fish and invertebrate bryozoans. And here we can see the life cycle of this Tetracapsuloides bryosalmoni. And so the infected bryozoans, they release the spores into the water and they infect the fish and they enter through the gill epithelia of the fish and they develop in the kidney of the fish, and the infected kidney release the spores by the urine into the water and they infect again the bryozoans, and also the aquatic worms are a vector for this parasite. And this disease was the mysterious, and in 1924, a German lady scientist, Marianne, she found first clinical sign in rainbow trout, and she wrote a book that is called Practicum der Fischkrankheit. It means a practical book for the fish disease. And she called amoeboid-like organism, amoeboid infection in their near. And that time it was called PKX, and then it was discovered as a proliferative kidney disease in salmonids. And this is basically the chronic inflammation of the kidney, which is characterized by profound dysregulation of B cell and T helper cell and decrease of myeloid cells. PKD is a temperature dependent. We can see the infection mainly the summer and autumn months, and normally we see the clinical sign if the water temperature is more than 15 degrees. And it causes high economic losses in the trout industry. We have this very old data, around 2.5 million pounds per year in the UK as per the FAST. But now it may be more. And now we are knowing that wild trout population in European Union is declining, and there are several reports from Austria, Germany, Norway, and Denmark also, and they are increasing day by day. And PKD outbreaks also occurred in wild mountain whitefish in Yellowstone River in Montana. And if we see the geographical distribution of this parasite has been reported from wild and farm fish from the North America, and also in some part of the Europe. And also it is reported from the European Union, but we have also trout culture in some part of the world, but there is no report of this parasite. If we see the clinical sign of the disease, we can see the renal hypertrophy in the infected fish. If we see the histology of this infected kidney, we can see pre-sporogenic stages, and also the immunostaining of this parasite, and also the parasite in the blood. And if we see the transmission, that the brown trout release the viable spores and it infects the bryozoans. However, the rainbow trout cannot transmit the parasite to the bryozoan. So recently, we have done NGS analysis for both of the hosts, and now we are analyzing the data, what kind of genes are responsible for these hosts. And we also found in our study that the brown trout is the carrier for this parasite, and it recovered from the disease even after 5 years post-infection. And this fish is also able to release the viable spores up to 5 years post-infection. And now we want to know what is the bryozoans. They are colonical suspension feeding sessile invertebrate, and they grow by a budding of zoids, and the optimal growth is 18 degree, and they are submerged surface like the stones, plastic, and the tree roots. And Epsultana is the most common invertebrate host for this parasite. We also found vertical transmission of this parasite in bryozoans. So here we can see the parasite stages in the statoplast. And here we can see how we collect these statoplasts from the colony. Here are the laboratory setup that we established. And that is the hatching stages, how we hatch in the lab to maintain the life of this parasite between two hosts. And we can see how we cultivate in the lab. First, we extract the statoplast, and then we keep in a media to hatch them. And then we feed with the algae, and then we rear them under the laboratory conditions. And here is our laboratory cultivation setup in Vienna. We established a very good model for this parasite. And here is the cohabitation of bryozoans. We keep the infected fish with the bryozoans for 2 weeks, then we have the infection of the parasite. And here is a very good video. I don't know if it will work or not. So we applied in vivo induced antigen technology to identify pathogen antigens that are immunogenic and expressed in vivo during host infection. And we can see a schematic diagram for in vivo induced antigen technology. We collected sera from the infected fish and also the parasite. And we have done serial absorption of the sera with the parasite antigens with different forms. And we collected this interest antibody, and parallelly we have done mRNA of the parasite and cloned in lambda triple X2 vector, and this phage expression library were screened using the immuno screening of the sera. And we identify the positive phases, and we have done sequencing, and they were validated by real-time PCR. And here is the antibody determination of the pooled sera by the indirect ELISA with different forms. So here we can see white-blue screening on LVMG agar plate. So white arrow indicate the recombinant plague, and the blue indicate the non-recombinant plague. So we selected only the white arrows indicating a recombinant clone, then we removed from the gel, and we identify and by the sequencing. And overall, we identify 136 in vivo induced antigen of this parasite, and they were basically involved in signal transduction, cytoskeleton, transport, and metabolism, so on. And we can see the biological process of these genes in a different form. Also, we can see the molecular function of these genes. So here are first 4 top in vivo induced parasite genes like calmodulin that is involved in multifunctional intermediate calcium binding messenger protein, and also the race-related RAB35 and transmembrane also an effect in capping protein. And we can see the expression in the infected fish and also the infected bryozoans. We only found the expression in the infected fish, not in the infected bryozoans. So our study improved understanding the virulence mechanism of this tetracapsuloids bryosalmoni, and these data will help to development of vaccine and novel control methods for this disease. Based on our finding, our team in Vienna, they are developing a vaccine against this parasite. And additionally, we have to do temperature-dependent global transcriptome data are needed to provide detailed information on the regulation of virulence factor of this parasite at what temperature they are expressed. And I would like to thank Badminton Vienna and also our boss, Professor Ilmat Bauli, and our international partner, Professor Jerry Bartholomew, USA, and Professor Nielsen Frank from Norway, and Bernard from Germany, and Vienna Vice Center. And finally, our director from SEMA FARI for giving me a table grant to join this conference in Chennai. Thank you.
Thank you. We have time for a couple of questions.
Hi, very nice presentation. These genes that you showed in the table,
Yeah.
they are the genes expressed by the virus, by the parasite?
Yeah, these are immunogenic and also expressed in the infected fish.
Okay. So you are hoping to develop a vaccine using these?
Yeah, so our team is working basically on the calmodulin. This is also reported from other animal parasite like Toxoplasma gondii, where they also found this calmodulin is also the immunogenic and the pathogenic for the pig. So we will also do such kind of things. So some of my friends, they are doing, and it will take some time to develop the vaccine.
But do you think that trying to find an antigenic gene is not really the same as trying to find a protective gene?
Yeah,
so
they are,
protective antigen,
because
whether it is
they are immunogenic. It means they may be the
may be protective.
protective. Why not?
Are there any more questions? No? Okay, well, thank you for your presentation. Oh,
Next presenter is Pushpa Mitra Panigrahi. She'll be presenting engineering 2D nanosheets for point of use biosensors, selective identification of volatile organic compounds emitted by stress shimps.
Good afternoon, everyone. So I'm Pushpa Mitra. So first of all, I would like to tell you that for last 3 days, I'm here in this symposium. I can see all of you, most of the people here are from biological background who are directly or indirectly concerned about health management of aquatic species. But I am from material science dealing with nanomaterials. So I can see this list explode in this area. But what my intention is, at the end of the talk, I can convince you that material science as a fingerprint can put in your domain. So coming to why the material science, when this is low-dimensional material, we are interested in this aquaculture because it is connected with nearly 7 to 8 SDG goals. But the problem is nanomaterials, like material science, is least explored. So when we wanted to come to this domain, we tried to explore what is this aquaculture when it is mostly dealing with shrimp. So it is a high-value $1 million industry, of course, but it has its hurdles when this aquatic species like the shrimps are stressed. It could be with viral infection or bacterial disease. And the loss is humongous. It's morning, from one session I could hear that is 8,000 crores. It's really huge. So then we come to the picture where we can come to this picture. One of the things is early detection if we could do. And this early detection, when it comes to biological science or in any field, it's not completely new. Material science has strong foothold when it comes to detection and dealing with nanomaterials, of course. So first, why we were very comfortable and confident that we can come to this field. So we have, like if you look at the volatile organic compound, I think many of you will be knowing about volatile organic compounds. These are carbon-based materials, but they produced during the cellular process. So they establish a metabolic fingerprint. So how it comes, like every human being, when we inhale oxygen, and we commonly know that we exhale carbon dioxide. But along with carbon dioxide, we also, in our exhale breath, there will be volatile organic VOCs depending upon human being's health condition. And mostly it is comes to the picture and it is very highlighted when it is the person is having cancer. So depending upon that, there are nearly 1,000 volatile organic compounds we have to deal with. And one of the example is, if it is person is affected with lung cancer, so far we are working in collaboration with medical scientists. So they tell there are 16 VOCs which are very specific to lung cancer. So then here comes, if you have developed a sensor and you can identify the volatile organic compounds, these VOCs, in your exhale breath, then the changes is really drastic. You don't need to, the person need not go for this painful biopsy procedure. And then it is always told that to deal with cancer, prevention is the best care, and much before. And what the emphasis is, when the cancer starts, the metabolic process in the cell starts, it's even much stage 1 that can be detected. So here we have been working, but the thing is here, we have to deal with nanomaterials when the system is coming to such precise scale. You are dealing with one molecule even. So then I will come back, what are these nanomaterials? This is the one for last couple of years we have been working. So this is the paper we published. This is dealing with 2D nanomaterials. That I will also come. Then we also worked, another paper also we got, because this is all with lung cancer, where 16 volatile VOCs are need to be identified, and there is a recent paper also we came. So what I wanted to tell here that we have some background to deal with this VOCs. We are not completely new. Now, I want to tell that entire world, the research field, one point we are together when we are dealing with this periodic table where entire universe is with maximum 118 elements. And your field, you are dealing with some elements, and we are dealing with some elements. But here comes the nanomaterials, why it is so wonderful. I just want to give a small example, because last 3 days, I see many people are just know that nanomaterial is amazing. But how amazing, I just want to give a small example. Like let's consider only one of the things. Gold. Everybody likes gold, I think, because it's beautiful and it is very expensive. But when it is the material scientist, we don't appreciate it much because it is chemically inactive. It doesn't help much. But this gold, if you want to nano-size it, it's beautiful, but it's become differently beautiful. It's no more that yellow gold. But this becomes chemically very, very active. And then it has a big fingerprint in many fields, coming to drug delivery and cancer and many things. But I want to make it clear that when it is nanosensing, it is not that effective. But it is one of the very good examples I would like to tell. Then comes when this material in the periodic table, you look into that and you try to engineer, then you get 2D nanomaterials. One of the best thing is in this 2D field, how it came, nanomaterials, when you got graphene. So graphene, all of you are now holding graphene in your hand because the phone, mobile phone you are holding and you are doing tak, 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 messages you are changing, that is the background is graphene is there. And for that, the Nobel Prize came in 2010. And after graphene, things got changed. You got many nanomaterials got designed. Then what comes when you fit this nanomaterial in the electric circuit for sensor? So in sensing field, these nanomaterials have huge impact. There are different kinds of nanomaterials. Once you deal with the periodic table, you can synthesize, depending upon your requirement, these nanomaterials. So once you connect them with the electrodes and allow the gas to pass through, then sensing can happen, and it is ultra high sensitive. It can detect in a molecular scale. Then the second thing is, it can identify different gases at the same time. It will give different signals. And the third thing is, they are stable and reproducible. So cost management is also there. So with that, so thinking this is the background, this is the objective of our research when we started. So we thought why we cannot use these nanomaterials to handle this shrimp when they are stressed. So here I have a small video. It is just to give the glimpse that what can be done using nanomaterials. First of the things, you have to identify, we have to identify when the shrimps are stressed, what volatile organic compounds they emit. And then how it can be identified so that we can detect before the disease comes. So this here comes to the materials. So this is the, when we come to nanomaterials, things changes completely. And here, the material atoms, everything goes, who plays the dominant role? They play the electrons. And these electrons, which Einstein had hard to believe, that electrons are no more particles, they are waves. So, dealing with nanomaterials, you have to deal with electrons. And that time, it is not easy to synthesize this material. It is expensive and it is time consuming. So computational simulation has a strong hold in predicting these nanomaterials which can handle this gas sensing purpose or any other energy storage purpose or any other. So depending upon your system, you have to, I think I'm wrong. So this is the DFT, the mathematical background also got the Nobel Prize. With that we started. Then this is how first we started our work with tungsten diselenide nano where we modeled. And we identified when the shrimps are under stress, they produce this, there are several VOCs will be there. These are the VOCs they will definitely produce. Along with that, glucose, it is not a VOC, but it is a parameter. So with that, this is the result. Once the VOC you bring close to the nanomaterial, so they will give the signals because they will have an interaction with the cloud of electrons which is on the surface of nanomaterials. And if your system is not that efficient, you can further engineer with dopants, suitable dopants. So here we have several dopants like nickel, As, cobalt, iron, and gallium. And this is, we found, the nickel is one of the things which can give the better result. Then we calculated the sensitivity. But one of the catch here is most of the nano sensors work in air medium. So here comes the water medium, where our normal seat was not efficient, but once it was doped with nickel, it became very efficient. Then this is the recent paper we published. And I want to show you how this nanomaterials looks like. If you go to TEM or SEM, you can see their picture. And below the pictures are that material's lifeline, how the electrons behave. And once you tune the electrons, you can tune the entire engineering systems. So this is how it shows the variation when the nickel is doped to the system and when the VOCs it starts interacting with. So then the system is connected as like I was showing you the cartoon with the electrodes, and we can see that one of the VOC TMA, this nickel doped WaC2 is very highly efficient. Then there are several other factors. We have started with Maxins. This is the new generation material that we doped with several other materials. This is one of the good efficiency pattern we can show. And then it is the recovery time, because if you use the nanomaterial and if they strongly absorb it, you cannot reuse. For that, you can see that their recovery time is in the second label. So it can be reused, and it is also highly efficient in presence of other interfering gases. Because when the VOCs are there, they will be always along with other gases. So that efficiency needs to be checked. So these are the work so far we are dealing with. Then it is a long way to go. Once the nanomaterial is being synthesized and it is being identified and the practical applicability will be there, then we can go with miniaturization is there, low-cost manufacturing is there, then IoT integration is there. It can come really to your picture, your domain, and soon I think the practical applicability in aquaculture, it will be there. Here I would like to, at the end, introduce my university. I am from Hindustan Institute of Technology and Science. I lead a research center. It's called Center for Clean Energy and Nanoconvergence. And it was inaugurated by Dr. APJ Abdul Kalam. All of you know him. And the person who is very tall here, I worked with him. He was Professor Borio Johansson. He was the Nobel Prize Selection Committee head when I was there. And this is my research team. And this is the group with whom we work. And at the end, I would like to thank the entire team who gave me the opportunity to present here, and also the audience. Thank you.
Thank you. We have time for one very quick question.
Yes.
Very nice, and something different from all the presentations here. In a real time, can we have sensors installed with these things and then predict diseases? How it can be incorporated with AI technologies? Is there any?
Yes. We have an underwater team also. There is a group of engineers there working. So they are using this AI technology, but it is not yet with aquaculture for sure. But there are for different field they are working. And for cancer research, it is very well, now the research group is very well active.
Thank you.
Thank you. I'm sure you're going to have lots of really interesting conversations over dinner. Apologies, we haven't got time for any more questions.
You told that some VOCs have been identified for cancer. Similarly, can we identify some VOCs for WSSV?
VOCs for?
WSSV.
For that actually I am here. That's what I was thinking. If nobody asks me questions, then maybe it will not be appreciated. So we are in constant term. We came with our group of people to you, that you, the people here, will help us identifying those things first. So this few VOCs we identified, of course, with your group, because the idea is when the shrimp is completely dead, they emit lots of VOCs. And whatever the sensors are there in the market, you can do that. But when they are affected, getting affected, they are starting to get stressed, that time the amount is very, very less. So that time the sensor will be useful. But of course, I spoke to someone here, he was asking how to identify. I myself want to know if it is a viral affected or it is a bacterial affected. That, of course, we really don't know. We will come and knock your door, like how to identify those VOCs. So that's why we should work together. So time has come, so material science can come to biological science.
Thank you.
Thank you.
Thank you so much. Our next presenter is Makesh Marappan. Is there? He'll be presenting markerless expression of nervous necrosis virus caspid protein in microalgae. Please.
Good evening, ladies and gentlemen. So I'm Makesh. I'll be presenting my work on markerless expression of nervous necrosis virus capsid protein in microalgae. So fish represents 16% of all animal protein consumed globally, and this percentage is likely to increase in the coming years. And of course, aquaculture is poised to meet this demand by diversification and intensification. But there is a major impediment to this in the form of diseases, which causes economic loss and production loss, and of course 15% of the value is lost because of the diseases. Prophylaxis through vaccination can offset most of this loss due to diseases. The Norwegian aquaculture scenario is evident with this. The introduction of vaccines in 1990s, early 90s, has resulted in the reduced antibacterial consumption, whereas the production has gone up drastically. So, but most of these vaccines introduced or the presently what is available in the market, they're all of injectable in nature, and there are a lot of difficulties, practical difficulties in administering this vaccine to the aquatic animals, especially for the smaller animals. But oral vaccines on the other side has which can overcome the difficulties of the injectable vaccines, especially for the smaller ones and for the mass vaccination. Oral delivery of fish vaccines can be done using many ways. Maybe morning, Dr. Azad was discussing about the oral delivery of vaccines through probiotic bacteria. There's many other ways also, but there's another way of delivering it through microalgae. This has a lot of advantages over other oral delivery, which we'll be discussing. This microalgae is basically, and most of the work has been done with this freshwater microalgae, Chlamydomonas reinhardti, which is a motile single-cell green algae. And this has been widely used in industrial biotechnology for biofuel production and also for producing recombinant proteins, therapeutics, and antibodies, etc. And this is actually generally recognized as a safe one, along with some of the microalgal species. And the production of this mass production is very cheaper because it requires only inorganic chemicals for mass production, and it is phototropic. So, and also it has, since being a eukaryote, it has all the post-translational modifications, and can also be lyophilized and stored for at least 6 months. So you don't have need to have a cold chain for storing these vaccines. And this biomass production, as I told you, it's very rapid, and the entire biomass can be used in the feed, incorporated into the feed and applied as a vaccine. So there's no downstream processing, so the production cost and delivery will be much cheaper compared to other forms of the vaccine, other forms of oral delivery. And this antigen can also be expressed either in the nucleus or in the chloroplast. But chloroplast expression has some more advantages because chloroplast itself is it's a self-contained organelle having its own genetic system, and we can insert this the gene of interest precisely into the defined location into the chloroplast genome. And also, it doesn't inside the chloroplast there is no gene silencing mechanism, so the production of recombinant protein is ideal. And you can also use it for a multivalent vaccine. Many genes can be incorporated. And also, the protein gets accumulated in the chloroplast. In case if you want to purify it, it is also quite easier. And also, in the chloroplast, actually in the microalgae, there is a single chloroplast, but there are many copies of this chloroplast genome. So obtaining this homoplasmic transforms chains is much easier compared to the plants where the number of chloroplasts is high in number. So coming to the pathogen on which we are working, in the viral nervous necrosis caused by nervous necrosis virus. It's a very serious virus of brackish water fishes affecting almost 175 species, including some marine and freshwater fish also. Basically, there are 4 genotypes, and RGNN, the red spotted group of nervous necrosis virus, is the most widely prevalent throughout the world, especially in the tropical waters, including India. And it affects mostly the larval and early juvenile stages, causing 100% mortality, whereas the adult fish, when it gets infected, they become carriers. They don't die, they don't exhibit symptoms, but they pass on the virus through the eggs and milk to the offsprings, causing huge mortality. Vaccination is the only practical way to prevent this vertical transmission, and also horizontal. And this virus has a very small virus, has a single structural protein. The RNA2 codes for the capsid protein, which we are going to use it for the vaccine production. So this is the mechanism how which we are going to develop the plasmid constructs for transforming the microalgae, for which we codon optimize the NNV capsid protein gene for expressing in the microalgae. And we amplify this gene with the primers having the restriction sites, SAP1 and SPH1. Restriction enzyme digested with these two enzymes, purified it. And similarly, the plasmid also was, which also has the 2 homologous 5' and 3' homologous region, which is similar to the regions or the same sequences present in the chloroplast genome also. And this has also the same restriction sites, so it was restriction enzyme digested, purified, and ligated, and transformed into E. coli. And also, this plasmid has an ampicillin resistance gene, so this ampicillin resistance was used for select the transformants. And these are the gel images showing the positive colonies. So this plasmid which we have constructed was used to transform the Chlamydomonas rhabdoviridis. The Chlamydomonas, the genome, of course, it's 200 kbs. The selected region it has this ADA gene. Actually, the PSBH gene, which is responsible for the photosynthesis 2, has been replaced with this ADA gene cassette, which is shown in the red circle. So that means this phototropic nature has been removed, and the anti-spectrum, which ADA gene, which is codes for the spectronamycin, has been introduced. So this strain, this mutant strain, which lacks this PSBH gene, cannot grow in the presence of light, and it requires acetate-containing media for its growth. So this strain is grown on TAP media, tris acetate phosphate media, containing exposed to in the presence of spectronamycin, so that the mutant strains can be selected. After the transformation with the plasmid constructs which you have developed, this between the 2, 5' and 3' homologous regions, the genes present in the plasmid gets inserted into the host. So this, after the transformation, the transformed cells regains the PSBH gene along with the gene of interest. So this regains the phototropism, and the spectronamycin gene is lost. So these mutants, the transformants can be selected on acetate devoid media, that is the HSM, high salt, minimal medium, devoid of acetate, but exposed to light. So only the transformants will grow, whereas the untransformed cells cannot grow because of the absence of the acetate. So we use two different strains, TN72 and HT72 for transformation. Only difference between these two is the TN72 is devoid of cell wall, whereas HT72 has the cell wall, but rest is all, both are acetate requiring light sensitive mutants with spectromycin resistant. Transformation also we did by electroporation and glass bead aberration methods, and electroporation with the square wave method seemed to be more appropriate. It yielded more colonies. And after the transformation, the cells were plated on high salt minimal agar plates and incubated overnight in dark, and subsequently under medium intensity light at 26 degrees centigrade. The colonies develop very slowly. It takes at least 3 weeks for the colonies to develop, and once the colonies develop, we restrict them on HSM agar plates and HSM broth. You can see the minute colonies in the plates on the lower side, which takes at least 3 weeks for growth. Whereas the normal untransformed cells do not grow in this media, which is shown in the other plate. So to confirm the positive clones, we did a PCR confirmation. The first gel image shows the PCR for the RBCL gene, which is nothing but a housekeeping gene present in the transformants and also the mutant strains. So just to show that the DNA isolation, everything is working well. The second gel image is for the PSBH gene. So it shows that the PSBH gene, which is not present in the mutant strain, original mutant strains, has been replaced. They have regained this PSBH gene, because of which they become phototropic, whereas the untransformed cells do not have this. And the third gel image shows the gene of interest, which is incorporated. That is also, again, present in the transformed cells, whereas not in the untransformed cells. We also, this is an ongoing studies, and we did a preliminary study on the expression of this protein also, for which we did a Western blot using polyclonal antibodies, where we could see the expression of a 40 kDa capsid protein, whereas the untransformed cells did not yield any bands. Of course, there was some non-specific bands also, and we are optimizing the expression of these protein, and post which we will be going for vaccination trials. So concluding, so the industrial biotechnology sector is currently relying on mostly heterotrophic organisms like bacteria, yeast, and mammalian or insect cells for recombinant protein production or biosynthesis of many of the compounds. But the recent success of phototropic microalgae for biosynthesis of therapeutic proteins has discovered its potential as an alternative platform for the commercial production of many of these compounds, including recombinant proteins, which can be used as along with the cells, can be used as a vaccine delivery mechanism for fish. And this has an added advantage that there's no antibiotic markers in the final transformant, so this becomes easy for the regulatory approvals to get it through. And if this success, and if it can elicit a good immune response and protective immune response, this is going to transform the way which we are going to vaccinate our aquatic animals. So I would like to acknowledge my research scholar, Mr. Arul Kumar, and all my colleagues who have helped me in this work. I also like to thank my director, Siba, for providing all the facilities and support for the work. And I would like to thank CRP and Vaccines and Diagnostics for providing the funding support, and the organizers of DAA for giving me an opportunity to present my work here. Thank you.
Many thanks. Do we have time for some questions?
Hi, very nice presentation. Any work on the localization of the recombinant protein which is expressed in the algae, and whether it is in active form or not?
Actually, localization we have not studied so far, but as per the literature, if we insert the gene into the chloroplast genome, it gets localized in the chloroplast only. It is not released into the cell or in the chloroplasm or the cytoplasm of the cell. So it should be localized within the chloroplast. And the biological activity, of course, the Western blot shows that it is the antibodies raised against the capsid protein is able to bind, so it should be biologically active. But we need to do vaccination trials in fish and see that they are able to produce antibodies which can bind to the virus or neutralize the virus. That study is yet to be done.
Is there time for one more? Yep, here we go.
Sir, this can be done only in Chlamydomonas or any other marine microalgae is possible?
Pardon me?
Whether it can be incorporated in other marine microalgae like Nanochloropsis or
Yeah, it can be done. But right now, there are these mutant strains, which I was telling, the Chlamydomonas raynaudii TN72, HT72. So these are all designed for this recombinant protein expression. But for marine microalgae, there is, of course, a lot of work is being done, but such strains are not available. Of course, we are also thinking, we are planning to develop such strains of Dunaliella and all for expression of recombinant proteins, so that can be used in the marine sector also.
But here we have to focus on the large-scale production of Chlamydomonas also for vaccine production.
Yeah, definitely. Yeah, and of course, that is very easy in aquaculture sector. It doesn't need any big laboratory setup to mass produce this microalgae. All we need is some tanks where even every fish or shrimp hatcheries have the live feed section where they grow this microalgae to thousands of liters. So it's not a big deal to produce in mass of this recombinant thing. Only thing, before that, it has to go through the biosafety procedures, should be cleared by the ethical biosafety committees for field applications. Once done, it can be done, easily produced in any hatcheries.
Thank you.
Thank you. I think we'll move on to the next speaker now. Thank you very much.
Thank you.
Our last presentation of this session is by Jenna, How Tilapia Macrophages Adapt Functional and Epigenetic Insights into Polarization and Trained Immunity. Please.
Thank you, ma'am. So good evening, everyone. I'm Gina, and today I'm here to present my work on macrophages and trained immunity that we have done as a part of our project in vivo, but here I'll be presenting the in vitro part of it. So the title of the work is How Tilapia Macrophages Adapt Functional and Epigenetic Insights into Polarization and Trained Immunity. So health management in aquaculture plays an important role, and it basically works in two aspects. Basically, the two major strategies that are involved is either by targeting a specific pathogen or by improving the overall health of the animals. So improving the overall health of the animals is basically made or done through the application of immunostimulants or probiotics and prebiotics, etc. Whereas the specific pathogen is targeted by enhancing or improving the adaptive immune system, which is by the processes like vaccination. So here, basically the immune system of fish or the vertebrates possess two arms, the innate immune system and the adaptive immune system. So the innate immune system acts in a rapid way, but it is non-specific in action. However, the adaptive immune system plays a major role by providing memory to what encounter it has made, and also it adapts the individual in order to respond to a specific way. So here, if I say the innate immune system also possesses a kind of memory that we can make use of, and it provides the memory in a broad spectrum way. So that is called as innate immune memory or the trained immunity. So the trained immunity is a concept recently introduced by Nithya et al. In 2011, and the works with trained immunity or the innate immune memory in fishes is at its infancy, and it is being progressing. So here, in the case of innate immunity or the innate immune memory, the major players are the macrophages. And the macrophages, as we know, have important role in phagocytosis, and they have different capabilities, which we'll be seeing in the coming slides. So they have the ability to polarize, and also they show plasticity. That means polarization means based on the microenvironment that they are in, they take up different phenotypes, like it can be pro-inflammatory macrophages and anti-inflammatory macrophages, which result in healing. And the plasticity means by providing different stimuli that what we need, whether we want to heal or we want to create inflammation, so we can reprogram the macrophages as well. So this particular trained immunity involves the use of, basically the players are macrophages, as I told, and it is basically T and B cell independent, and so it provides a non-specific protection. So there are a lot of importance for trained immunity in the coming days, because it can provide broad spectrum protection, long-term immune protection, and also we can make use of them in the vaccine adjuvants and in case of early life immunization, which will be very important in the case of fisheries. However, many studies have been done, but however, in the case of polarization and plasticity of macrophages in fishes, the studies are very minimum, and also the interventions for harnessing innate immune memory in aquaculture health management are yet to be taken up, and we have started our work, and let's see what are the results. So the first, for addressing that, or to meet the gaps, the first experiment what we done was a cell characterization. For that, the macrophages were isolated from tilapia and were cultured and were stimulated in order to characterize them using different doses of lipopolysaccharide. So the characterization was done by immunofluorescence using the IBA1 protein, and by quantifying the innate immune markers and also the phagocytic activity. So first, let's see the morphology of the cells. So the macrophages, culturing them are entirely difficult, and somehow we were lucky to we were fortunate to get the macrophage culture, and initially they were like, as all the cells, like they were floating and they were not attaching and all, but once they started adherence, we could do the passaging, and yes, we could see the pseudopodia formation and all, and these cells were used for the work. So here the mononuclear cells were characterized, as I told you, by localization of IBA1 protein, and the ROS production, the NO production, and the percentage phagocytosis in the stimulated cells, the upregulation in the stimulated cells has helped us in characterizing the cells as the macrophages. Now, the second experiment was to functionally characterize the cells. So the cells, we wanted to do the polarization of the cells, so they were treated with different ligands, that is LPS, lipopolysaccharide, beta-glucan, and peptidoglycan, to polarize them as M1 macrophages, and CAMP, cyclic AMP, was used for polarizing them into M2 macrophages. And also the plasticity studies, that is whether they are shifting from M1 to M2, that attempt was made. And further, the morphological analysis, functional assay, and the expression analysis of expression profiling of M1 markers, INOS, IL-1 beta, and M2 markers, arginase 2, TGM2B, and TIMP2B marker genes were profiled. So coming to the morphology of the cells, there were little difference, like basically the pro-inflammatory macrophages were showing a bit larger size, and also the pseudopodial formation was too much. But here the pictures are not so good, but however, we proceeded with the work, and these are the functional assay, results of the functional assay, which shows that the as I told you, ROS, nitric oxide synthase, was quantified for the M1 macrophages, and for the M2 macrophages, the arginase 2. So the group which was treated with LPS, lipopolysaccharide, has given the highest ROS production, and the NO was highest in the case of peptidoglycan, whereas beta-glucan, we couldn't find that much of higher significant results. However, the percentage phagocytosis was high in case of beta-glucan-induced cells. And the M2 macrophages has given the arginase production higher. Now, coming to detection of polarization, to to supplement that production of NO, ROS, etc., we have done the expression profiles of the M1 markers, that is INOS genes and the interleukin-1 beta, which has shown elevations, significant elevations in case of LPS and peptidoglycan when compared to the beta-glucan. Now, the M2 marker, the arginase 2, metalloproteases, and the TGM2B that we have done, and that also has given promising results. Now, when we did the macrophage plasticity, that means shifting from M1 to M2 stage, so here we could clearly see in the case of LPS stimulated ones, LPS and peptidoglycan stimulated M1s, there was a clear shift. The arginase production was more. As seen in the previous slides, the beta-glucan was little less. Here also, the beta-glucan has given less production of the arginase. So now, to support that, the M2 markers were also done, the expression profiles of the M2 markers were also done using qPCR. The work has supported the results of the previous slides. Now, coming to the evaluation of trained immunity, basically the trained immunity, as I told you, it can be used for a broad spectrum protection. So here what we have done is we wanted to prove that trained immunity is existing, and we wanted to show that there is immune training happening in the cells. For that we used an inhibitor, that is metformin, which is a known inhibitor of trained immunity, and we classified the macrophages into 6 groups, that is control, beta-glucan stimulated group, beta-glucan plus metformin stimulation, and the lipopolysaccharide re-stimulation, lipopolysaccharide re-stimulation plus metformin, and the lipopolysaccharide alone group. And further, the immune marker evaluation, innate immune markers, epigenetic markers, and trained immunity markers were analyzed. So coming to the evaluation of innate immune markers, the ELISA for those pro-inflammatory cytokines were done. And here, the re-stimulated group, basically trained immunity, we have a priming and a boosting step, and that is the re-stimulation. So there, in the case of re-stimulated group, where metformin was not used, the highest expression was found. And the metformin, where the metformin was used, it was clearly evident there was no production of the pro-inflammatory cytokines. Now coming to the evaluation of the trained immunity markers, because trained immunity, the basic mechanism is epigenetic reprogramming and the metabolic reprogramming. So here the HIF-1 alpha and mTOR and percentage phagocytosis was done. And here the HIF-1 alpha, all those markers of the trained immunity has given elevated expression in the case of the re-stimulated groups. And the epigenetic markers, histone 3 lysine 4 methylation, histone 3 lysine 4 trimethylation, and histone 3 lysine 27 acetylation was targeted. And using ELISA, we have done this. And it's here in the case also, the beta-glucan stimulated group has given the highest expression. So the highest heightened expression of trained immunity specific epigenetic markers were found in stimulated and re-stimulated groups. So coming to the summary of this work, the primary macrophage culture was developed from Tilapia head kidney. For M1 polarization, the cells were stimulated with lipopolysaccharide, beta-glucan, and peptidoglycan, which are the known ligands. And for M2 polarization, the cells were stimulated with cyclic AMP, and the cellular responses were quantified. For plasticity assessment, the cells which were initially polarized to M1 were re-stimulated using the CAMP to induce the M2 repolarization. Thus, we demonstrated the development of an innate immune memory. That mechanism we demonstrated in trained macrophages, and the combination of stimulants and metformin actually can be used as a solid method to prove trained immunity in the cells. And with the outcome of this, when we compare with the in vivo as results, in vivo we could obtain the longevity of protection for about 4 months, and that too in the case of, that too a broad spectrum protection was offered. And thus, from this I conclude, innate immune training offers a new frontier for fish health management, driving sustainability, resilience, and next generation vaccine strategies, because now we are facing the problem of multiple pathogen infections, So in such cases, if we target a single pathogen, every time that may not be viable. So if we have a setup or if we have an approach that can target multiple pathogens, that would be of help to the farmers and also to the aquaculture system. So these are my references, and thank you everyone. And I place on record my thanks to the organizers of DAA12 for giving me this opportunity to present this work here. And also I thank ICR CIFE, director, and all my team members and my dear students for conducting this work. Thank you all. And this was not my final presentation. Actually, I uploaded a presentation, and this was the previous presentation, which I first uploaded long back. Anyways, I could present it. Thank you so much.
Thank you very much. Do we have any questions?
Did you do any, this one on the fixed macrophages and then the circulating macrophages? There are two things.
Yes.
Could you do anything on the fixed macrophages?
No, we isolated from the head kidney of the fishes. Yeah. Tissue resident.
Any other questions? Thank you very much.
Thank you.
So that brings us to the end of this first part of the session. So I'd just like to thank all of our speakers again. Thank you.